Thank you for that very nice introduction. I'm Johann Gudjonsson. I'm a dermatologist at Ann Arbor, Michigan, and I've been studying the pathophysiology of psoriasis and GPP for quite a while now. And the title of my talk is very excited to be with you today, and I'd like to thank the organizers for giving me the opportunity to show you some of the findings and the data that we have been working on. So these are my conflicts of interest. And just to kind of give you a brief overview of GPP. So it's a minor subset of psoriasis. It accounts for about 1 to 2% of psoriasis cases. Pustular psoriasis is separated into a few distinct clinical subtypes, including generalized types of pustular psoriasis. The most severe one is the generalized pustular psoriasis, the von Schumbusch variant, but there are also other variants such as annular pustular psoriasis and impetigo herpetiformis. And then also localized pustular variants such as pustulosis palmoplantaris and the acrodermatitis continua of Halapo. And GPP is interesting in contrast to other types of psoriasis, given its explosive onset and often these life-threatening complications that can arise from this disease. And you can see evidence of that from the clinical photos that I have on the right. So if you look at the histopathology of GPP, what I have here on the left is the histopathology of plaque psoriasis, and I thought that was worthwhile to have that to kind of contrast to what we see in pustular psoriasis. This shows you the transition in plaque psoriasis from uninvolved distant to uninvolved near edge, the plaque center, and then the active edge of psoriasis cases. And then here on the right, I have two images that shows you the typical histopathology of GPP. And there are two distinct but overlapping kind of features that can come with pustular psoriasis. One of them is we have the GPP superimposed on the classic kind of a plaque psoriasis kind of a background. The other one is the more spontaneous type where you get widespread pustules on practically otherwise normal appearing skin, where you really haven't had a chance to develop these epidermal hyperplasia that is characteristic of plaque psoriasis. We've made a lot of progress over the years in terms of understanding the genetic predisposition of psoriasis, and in particular GPP. As Dr. Capon kind of outlined in her lecture before, she kind of showed you some of the main genes that have been implicated in GPP pathogenesis. And you can see that some of it actually overlaps with that of plaque psoriasis. But the main ones that have been identified in GPP include the IL-36 receptor antagonist, the CARD14, the AP1S3, and then in 2020, we identified two new genes that predisposed us to pustular psoriasis specifically, the SERPIN-A3, which is a serum protease inhibitor, and then myeloperoxidase, a neutrophil-related gene. You can see that there's still being progress made in unraveling the pathophysiology and genetic predisposition of GPP. This is a complex slide, but again, this is where we have kind of outlined the pathogenesis of plaque psoriasis, where you have predisposing factors, you have disease initiation, and then disease maintenance. You have activation of the innate immune system and plasmacytoid dendritic cells, involvement of Th1 and Th17 cells, and then activation of this inflammatory cascade in psoriatic skin. When in plaque psoriasis, what really is different is that you get hyperactivation involving keratinocyte activation and proliferation and neutrophil infiltration, and it's the autoinflammatory response that is really dominating. And it's really neutrophils that dominate the picture in GPP. GPP is a neutrophil dominant disease. White just to plaque and particularly normal skin. You have massively elevated levels of this levels in skin. You can see it right here at the 1 to 10 microgram per deciliter. And then you have 1 to 10 microgram per deciliter in plaque. And particularly normal skin. You have massively elevated levels of this levels in skin. You can see it right here at the 1 to 10 microgram per deciliter in plaque. And particularly normal skin. You have massively elevated levels of this levels in skin. You can see it right here at the also get a lot of enzymes such as cathepsin G, neutrophil elastase, proteinase 3, neutrophil specific implication regarding activation of inflammation in GPP. So one of the key genes that have been implicated and cytokines that have been implicated in GPP pathogenesis is the IL-36 pro-inflammatory cytokines. These cytokines are a member of the IL-1 cytokine family, like I have over here. They're all closely related, so IL-36 beta, alpha, and gamma, and they all signal through the same receptor, the IL-36 receptor. That is also, it's a heterodimeric receptor that also has an IL-36 receptor accessory protein. All three of the IL-36 pro-inflammatory members signal through this receptor, and it's the receptor antagonist that can inhibit the signaling. And if you look at the expression of these cytokines in psoriatic skin, you'll see that it's markedly elevated in GPP compared to plaque psoriasis, and it's practically undetectable or close to undetectable in normal skin. This is a log 10 scale, and you can see the shift here. This is about 10-fold shift that we're seeing for both IL-36 alpha, IL-36 gamma, IL-36 receptor antagonist, and also to some extent the IL-36 beta. So all of the family members, including also the IL-36 receptor antagonist, are much more highly expressed in GPP compared to plaque psoriasis, and certainly more than you see in normal skin. And if you look and do transcriptomics of psoriasis, like I have on this slide over here, where we're looking at the gene expression that is being turned on, and again, we're comparing plaque psoriasis with pustular psoriasis. And on one end, we have an autoimmune response that is really dominating, and on the other end, we have an autoinflammatory response going on. And if you look at the biological processes that are being turned on in chronic plaque psoriasis, they all belong to the autoimmune arm of the immune system, such as adaptive immune response, the antigen processing, the presentation, type 1 interferon signaling, leukocyte-mediated immunity, MHC class 1 presentation, and a little bit of granulocyte chemotaxis. If we do the exact same experiment on pustular psoriasis skin, and we're looking at the genes that are being actively turned on, we get a completely different pattern than we see in chronic plaque psoriasis. We get more towards an autoinflammatory response, and it's the granulocyte chemotaxis and the antimicrobial humor response and regulation of monocyte chemotaxis that are really the dominant biological processes that are being turned on, processes that will turn on and bring in neutrophils into the skin. And if we look at the different cytokines, such as IL-36 and IL-17A, it's a striking pattern that we see where the IL-36, as you move towards the GPP or pustular subtypes of psoriasis, we get progressively higher levels of IL-36 expression. On the other end, IL-17A, which is from the adaptive immune arm of the system, the Th17 cells, that's progressively going down as we kind of get towards GPP, kind of defining these on the opposite ends of the immune spectrum. And the same thing if you use different types of tools. This is what we call like a gene set enrichment analysis. And what we're really looking at is the downstream genes that are being turned on by specific cytokines, and in this case, it's IL-36 cytokines. And we're comparing plaque psoriasis and with pustular psoriasis, and we're looking at the area, the red area over here of genes that are being turned on, or the area that are being suppressed and are IL-36 specific. You can see that there's quite a bit of an area in plaque psoriasis, both for upregulated and downregulated genes, that is highly significant. But if you look at the same response in pustular psoriasis, you get a much greater area, much greater significance for both upregulated and downregulated genes. Going along with the observation that IL-36, the GPP is truly an IL-36 dominant disease. IL-36 are also interesting that they're secreted as full-length proteins, and as such, they're actually relatively inactive. But it's the neutrophil genes that I showed you on the immunohistochemistry before, such as cathepsin G, proteinase 3, and elastase, that actually trim specific peptides from each one of the IL-36 family of cytokines and increase the biological activity of these by about 500-fold. So you can see that over here, the ones that are resistant to enzymatic digestion are shown here in red. The ones that are truncated are shown in black. And as you're going to get more elastase, you get much more pro-inflammatory activity and much greater release of IL-6 and IL-8. It's also quite interesting that it's not just the neutrophil proteases that play a key role in processing IL-36. It also is cathepsin S, which is a keratinocyte-derived protease. And if you look at it here in terms of IL-8 secretion, and with higher concentration of cathepsin S, you can really appreciate the processing of this is much greater with the cathepsin S compared to elastase or protease or cathepsin G from neutrophils. So what this indicates that it's keratinocyte-derived cathepsin S is actually a more potent activator of IL-36 compared to neutrophil elastase or cathepsin. And that indicates that it's not just the neutrophils that are critical in amplifying and turning on this axis, the keratinocytes themselves can do it as well. And if you look at where in the epidermis is this response happening, this is from single cell data that we use from psoriasis. We're looking at the genes and the cell-cell interactions that are happening in different layers of the epidermis. This is the supraspinous, spinous, and basal layer. And it's really within the supraspinous compartment of the epidermis where the IL-36 cytokine axis is feeding onto itself to amplify inflammatory responses in a self-sustaining manner. And if we look sophisticated like models, in vitro models to study the biology of IL-36, these are CRISPR-Cas9 knockouts of the IL-36 gamma and the IL-36 receptor in keratinocytes, and they're being stimulated with pro-inflammatory cytokines such as IL-17 and TNF-alpha. What you can appreciate is that you get a fairly robust response at baseline in the wild type lines, both for beta-defensin, IL-36 receptor antagonist, IL-36 gamma, and S100A7 pro-inflammatory genes. But if you knock out and block IL-36 gamma itself or the IL-36 receptor, you're really dampening down the response downstream of both IL-17A and TNF-alpha. So what does this all kind of mean? This is kind of a summary figure that I put together to kind of truly outline what the IL-36 axis is doing in GPP. So IL-36 can be turned on. It's downstream of pro-inflammatory cytokines such as IL-1, IL-17, and TNF. That increases expression of IL-36 in the keratinocytes. But it needs some kind of a danger signal such as ATP to really being secreted and released into the extracellular medium. It really has to be exposed to neutrophil nets, and particularly the neutrophil proteases, to get truncated and biologically active. And that increases the activity of these by about 500-fold. Cathepsin S from keratinocytes also contribute to this and can activate the IL-36 axis by itself. But the keratinocytes can also regulate this axis by secreting A-type serpins or serpin A's that actually inhibit this processing. The IL-36, the truncated IL-36, the biologically active component of IL-36, signals to the IL-36 receptors, activates the NF-kappa B pathway, and increases the expression of IL-36 even more. And if you look, and then that in turn increases expression of neutrophil chemokines such as CXCL1, CXCL8, and CXCL2 that brings in more neutrophils to really hyperactivate and amplify the process. And if you look at where those risk genes in GPP are located, they're all located around this same axis. So IL-36 receptor antagonists with the IL-36 receptors gene itself, CART14AP1S3 to kind of regulate the NF-kappa B pathway in keratinocytes, and then the myeloperoxidase in neutrophils that has been shown to have a role in regulating IL-36 processing. So looking at it in a more of a clinical way, so what's really happening in pustular psoriasis is this kind of a marked shift or adaptive immune response towards an autoimmune IL-17A that is dominating, but you have a component of IL-36. But when you move towards pustular psoriasis, IL-17A becomes much less prominent, and it's the IL-36 system or axis that is the most prominent and dominant in this form of disease. And that's what's driving the neutrophil infiltration and the characteristic pustule development that you can see in the skin of these patients. But just to kind of quickly summarize, so psoriasis, as I've shown you, exists on an inflammatory and clinical spectrum. In plaque psoriasis, it's the adaptive immune response that dominates. In pustular psoriasis, it's the innate response that dominates, particularly IL-36. The biology of pustular forms of psoriasis are still incompletely understood, although genetic findings provide novel insights, but a lot of work that remains to be done. IL-36 is a central cytokine to its pathogenesis, but we still have a poor understanding of the processing and inhibition of IL-36 cytokines. We really do not know what each one of those three IL-36 family members is really doing and which one is the one that is most important. Also, we know already, based on the studies that have already been done and published, that neutrophils play an active role in the inflammatory network in psoriasis, potentially and most prominently through activation and amplification of IL-36. But it's also the keratinocyte that can activate and also shut down IL-36 processing through cathepsin S or serpin A's secretion. So a very complex network of pro-inflammatory and anti-inflammatory responses that are happening within the supraspinous compartment of the epidermis. And it's going to be very exciting in the next few years to kind of really dig through this and figure out how the mechanism behind all of this actually is. So thank you for listening, and I'm happy to take any questions.